Welcome back to the Rapid Revision channel. 18-day series, 100% biology syllabus. Let's crush it.
Hello future doctors, welcome to day 10 of our 18 days NCERT Biology Rapid Revision Explainer. आज का टारगेट बिल्कुल क्लियर है. We're gonna absolutely master genetics part 1, specifically principles of inheritance and variation. You know, cancelled papers और recent NTA trends को देखते हुए ये यूनिट आपके RE-NEET 2026 के लिए एक टोटल गेम चेंजर होने वाली है. सो पूरा फोकस यहाँ रखना क्योंकि आज हम मेंडल्स लॉज से लेकर कॉम्प्लेक्स क्रोमोसोमल डिसऑर्डर्स तक ना सब कुछ आपके फिंगर टिप्स पर लाने वाले हैं. तो चलिए शुरू करते हैं.
Let's quickly scan our high-yield roadmap for today. First, we're cracking Mendel's laws with some killer mnemonics. Then we'll hit Mendelian exceptions, sex determination, chromosomal disorders, and finally, a high-stakes PYQ alert. हर एक concept आपके multi-statement and match the column marks confirm करेगा. Section 1, Mendel's laws and mnemonics. Let's get into the basics.
See, basics strong होंगे.
So we can essentially distill all of that complex crossing data down to Mendel's 3 core principles. First up, the law of dominance. This one's simple. In a heterozygous pair, one allele is just going to completely mask the other. Second, we have the law of segregation, meaning alleles separate during gamete formation. This is what gives us that classic 3-to-1 phenotypic ratio in a monohybrid cross. And third, the law of independent assortment, which tracks 2 genes to give us that famous 9-to-3 to 3-to-1 dihybrid ratio. Now, to keep all these straight under intense exam pressure, you can use the mnemonics AMIGO and PALM to really lock your ratios in. But listen, here is the major conceptual trap, so pay close attention. Mendel's law of independent assortment literally only holds true for unlinked genes. If genes are sitting right next to each other on the exact same chromosome, they're linked, they travel together. And that neat little 9-to-3 to 3-to-1 ratio, it completely falls apart.
ये तभी आप एग्जाम में कॉम्प्लेक्स पेडिग्री चार्ट सॉल्व कर पाओगे, राइट? To memorize Mendel's laws instantly, मैं आपको 2 superb mnemonics दे रहा हूँ, AMIGO and PALM. इन वर्ड्स को देखते ही ना आपको मेंडेलियन इनहेरिटेंस के सारे कोर कॉन्सेप्ट्स क्लिक कर जाएंगे. Remember, Mendel gave us 3 main things: the law of dominance, the law of segregation, and the law of independent assortment. These mnemonics, AMIGO and PALM, are literally gonna save you those crucial seconds in the exam hall.
Okay, now let's zoom in on the law of segregation. इसको purity of gametes भी कहा जाता है. और ध्यान रखना, this is absolutely crucial. Mendel's law of dominance has exceptions, but the law of segregation, it is the one law with 0 exceptions. No exceptions at all. Purity is 100%. Alleles separate completely during gamete formation without any blending whatsoever. Basically, एक gamete में हमेशा सिर्फ एक ही allele जाएगा. Section 2. Mendelian exceptions. Blending versus both.
Now this area, it's NTA's favorite trap for those tricky assertion-reason questions, simply because nature sometimes loves to break Mendel's rules. Look on the left, we have incomplete dominance. Think of Snapdragon or antirrhinum. जब आप red और white flowers को cross करते हैं, तो आपको अचानक से pink F1 generation मिलती है. That is pure, straight-up blending. But on the right, look at codominance. The absolute classic example here is the ABO blood group system in us humans. यहाँ blending बिल्कुल भी नहीं होती. Alleles IA और IB दोनों fully expressed होते हैं एक साथ, creating the AB blood group. It's like both are fighting for the spotlight and both of them actually win.
So what's really interesting here is the exact difference between the two. ये एक standard 2-mark NEET differentiation question है, guys. हमेशा अपना blend versus both test याद रखना. Pink is a blend, right? It's an intermediate phenotype which immediately tells you it's incomplete dominance. But the AB blood group? That shows both A and B antigens together, completely unblended. That's codominance. Literally just apply this blend versus both test and you'll solve these questions instantly without a single drop of confusion.
Section 3. Sex determination patterns.
Listen, identifying these specific patterns is almost a guaranteed match-the-column question every single cycle. Let's do a rapid-fire cytological breakdown. First up, the XXXO system, which we see in grasshoppers and cockroaches. Second, the ZZZW system in birds, like the peacock. अब यहाँ एक major catch है. ध्यान देना. In birds, the females are actually heterogametic, meaning they are ZW, and the males are homogametic, ZZ. Wild, right?
And third, for just a second here, and throw a question directly at you. This right here is a major recurring NTA trap that catches thousands of students every single year. How exactly do male honeybees make gametes? Take a second and really think about it. Well, if you said meiosis, you just fell right into the trap. Let's clear this up with the concept of haplodiploidy. In honeybees, females develop totally normally from fertilized eggs, so they are diploid. But the males, the drones, they develop from unfertilized eggs through a really cool process called parthenogenesis. The haplodiploid system in honeybees. यहाँ males haploid होते हैं because they're produced by parthenogenesis. और इसका सीधा सा मतलब ये है कि drone bees अपना sperm mitosis से बनाते हैं, meiosis से नहीं. Let's look a bit closer at this odd versus even chromosome split in insects like grasshoppers. It's so cool. Female grasshoppers have 18 chromosomes, that's an even number, representing XX. But the males, they only have 17 chromosomes, an odd number, representing XO. That O literally just means one sex chromosome is entirely missing. So a one chromosome difference within a single species, that immediately screams XX versus XO. This is a classic conceptual test of your cytological knowledge that NTA loves.
Section 4. Chromosomal genetic disorders. Okay, understanding these disorders is absolutely vital for the latest pattern of clinical scenario questions. We have to differentiate them clearly. Aneuploidy is just the gain or loss of one or maybe a few individual chromosomes. Think 2n plus 1 or 2n minus 1. RE होता है due to non-disjunction during meiosis. Polyploidy, on the other hand, is the multiplication of an entire genomic set, like going up to 3n or 4n. एक simple mnemonic याद रखना. Plus or minus a few equals aneuploidy, multiply a whole set equals polyploidy. By the way, polyploidy plants make coffee common, but animals may it's usually completely lethal. Let's rapidly drill the trio of aneuploid syndromes you absolutely must have memorized. Number 1, Down syndrome, or Trisomy 21. That's a 2n plus 1 condition leading to features like a furrowed tongue and a broad palm crease. Number 2, Klinefelter syndrome, XXY. This is a male with an extra X chromosome, which leads to certain feminine features like gynecomastia and also sterility. And number 3, Turner syndrome, XO. This is a female missing an entire X chromosome, showing symptoms like a webbed neck, short stature, and rudimentary ovaries. I really want to reinforce an expert strategy here. जब भी exam में disorders का question आए ना, first, separate the count change. Find out, is it aneuploidy? Once you figure that out, just map the specific chromosome error directly to the named syndrome. Trisomy 21 always maps Downs. 47 chromosomes with XXY, that maps to Klinefelters. 45 chromosomes with XO, Turners. Separate and map. Honestly, it's really that straightforward. Section 5. PYQ alert and NTA trends. All right, high stakes time. NTA absolutely loves linking classical genetics directly to molecular biology, and this classic previous year question proves it. In sickle cell anemia, what is the exact substitution at the 6th position of the beta-globin chain? Think about it. It's this tiny, tiny point mutation that somehow completely distorts the entire hemoglobin molecule. Can you recall the exact bases and amino acids involved? So the crucial takeaway here is the exact logic behind it. It is literally a single base change. The middle base, A, is substituted by U. This turns the codon GAG, which usually codes for glutamic acid, into GUG, which codes for valine. एक सिंगल लेटर के चेंज होने से ना पूरे रेड ब्लड सेल का शेप बिगड़कर सिकल जैसा हो जाता है. GAG to GUG, glutamic acid to valine. You need to lock this in your memory. And honestly, this brilliantly sets up a massive expert pro tip for you. Notice how valine is coded by GUG? Well, get this. Any codon starting with GU, whether it's GUU, GUC, GUA, or GUG, they all code for valine. Knowing this specific GU valine block lets you answer these complex codon questions instantly without having to memorize that massive full codon table. Remember, smart work beats hard work every single time.
आज का रैपिड रिवीजन कैसा लगा? We've covered the absolute essentials of genetics part 1. Apply these tricks, the blend versus both test, and the exact chromosome mappings in your mocks today. Master complex genetics easily with our structured revision series. Subscribe now for the rest of the 18-day schedule.
Hit that subscribe button right now
Hit
so
that
you don't
like
miss
button.
tomorrow's
Keep
high yield
revising,
breakdown.
and let's crush
Drop your
RE-NEET
doubts below.
2026. Before you go, think about this. If just one tiny A turning into a U can completely change the shape of your blood cells and your entire life, how fragile and perfect is the code that built you? See you in day 11.